Click for IB Biology, translational model at higher level. There's a blank PDF down in the description, along with a quiz of 10 questions with answers, and of course my AI prompt, which allows you to build multiple choice assessment in a chatbot of your choice. Students who are higher level students should first of all look at the standard level model before attempting this model. The two must be combined together. Unlike in the standard model, here we acknowledge that there are actually 3 positions on the large ribosome subunit, which allow the 3 sites known as the exit, the peptidyl, and the amino acyl. In the same way, the codons on the messenger RNA will attract the anticodons of tRNA molecules which already have the necessary amino acid attached to them. We know that in messenger RNA that the start codon for all genes is AUG methionine. So in our translational model, the first step will be that the tRNA molecule AUC will move into position on the peptidyl site corresponding to the AUG of the messenger RNA. With AUG site occupied by UAC methionine, the next part of our genetic code is UGU, this codon here, will attract in the anticodon ACA tRNA, and that is for cysteine amino acid. And of course, what's now going to happen is that a peptide bond will form between the two. The ribosome is now going to move one codon to the right to cover the CCU, which will then leave the methionine tRNA in this position here, the exit, and it breaks off and ejects the tRNA out into the cytoplasm here. Indeed, the energy between the methionine and the tRNA molecule is the energy required to build the peptide bond. Now with this tRNA molecule here in position, and now the next one, which is CCC, attracts GGG anticodon with proline, we're now going to form the next peptide bond between the second and third amino acid. And of course, then what happens is that the ribosome shifts one codon to the right. And now we have methionine, cysteine, proline, and the next amino acid is going to be arginine again. Notice that the tRNA molecule here, the energy has been removed to form this peptide bond, and that's then ejected, so that can now go off and find more of its amino acid. The next thing that happens is that the energy is taken from this bond here and used to build the next peptide bond, as you can see in this sequence here. And then the GGG tRNA is ejected. And so the ribosome is moving along in exactly the same way as we saw with our standard level model, except instead of 2 sites, we have 3 sites. And notice that the polypeptide chain is always attached to the P site. The E site ejects the tRNA, and this site brings in, the A site brings in the new, that's the A site there, that brings in the next amino acid. But the polypeptide chain is always attached to the tRNA molecule on the P site. And finally, our ribosome will actually arrive at the messenger RNA UAA, which if you take a look at the codon table, gives us a stop command. At this point, the tRNAs are all going to be ejected. The energy here is used to form this peptide bond here, and then we have the ejection of the polypeptide, which has the sequence methionine, cysteine, proline, arginine, and glycine. That's the primary structure, but of course this is already forming up into secondary and potentially with a long polypeptide chain, tertiary structures as well. These structures, these proteins, are subject to post-translational modification. Post-translational modification is the subject of our next video. Here's the subscription button to the channel and of course the playlist link.